Hello, and welcome to today's segment on biology, continuing with Module 2. Today's segment will be on energy systems. This is the fifth module in the segment. Hopefully you've had the chance to check out all the previous ones. We are going to be continuing today with a discussion on energy systems. Fundamentally, within the body, ATP, as many of you may know, is considered to be the universal energy currency of the body. What is ATP? Well, ATP stands for adenosine triphosphate. You can see the image on the right. You'll see that it is an adenine molecule, ribose, and 3 phosphate molecules attached to it. Now, the number of molecules attached to it, of course, changes the name. With just the base adenine and ribose, it's adenosine. With 1 phosphate added, it is AMP. With 2, it is ADP. And with 3, it is ATP. Now, we've already had certain discussions in the class where we've talked about how various levels of ATP versus ADP versus AMP regulate the activity of certain enzymes responsible for energy production. Most notably in beta cells. The regulation, the amount of AMP versus ADP versus ATP actually regulates how insulin is produced and secreted. So, how does it work? Well, each one of these phosphates that's bonded to the adenosine base molecule is extremely high energy. If you look at the configuration, now, those of you who may remember from chemistry, and we can certainly discuss this later, the bonds, actually bonding energy, connecting molecules together either requires energy or releases energy. And that has to do with kind of the energy state of the molecules both before and after. Now, attaching molecules to the adenosine base requires a significant amount of energy, so releasing them releases energy. But of course you say, well, what is this? Is it heat? What is this magical energy that's released? What does it actually involve? Well, what it actually involves is these phosphates. Now, the energy doesn't just dissipate when they break off. It is converted because these high energy phosphates move from this ATP molecule to other molecules and change their position, change their function. Examples. If you look at an enzyme, there are enzymes in the body that are essentially activated by ATP. In other words, they are switched on. What happens is one of these high energy phosphates is actually broken off the ATP molecule, and when it hits the enzyme, that phosphate actually pulls the molecule and changes its configuration. So you could say, well, there you go. ATP, this energy is used to activate or turn on an enzyme, or in some cases turn it off. Another case is channel or carrier proteins. One of the things we've discussed in the past when discussing cell membranes is that ATP, when it actually binds to a channel or carrier molecule, protein can actually open or close it. This is very, very critical since we know secretion, cell polarity, so many other things are controlled by these channel molecules. Think about neurons, think about action potentials and ions crossing back and forth and creating this energy gradient. Well, to create an energy gradient or to create a charge in these molecules, you need to effectively snap shut some of these carrier proteins and allow a charge to build up. ATP snaps these shut and releases them. This is how cells generate this sort of impulse. Muscle contraction is another case, and one of the ones we're most familiar with. Later on in this discussion, and there have been, we've certainly discussed in a little bit of segments of biomechanics, how muscle structure, how muscle proteins actually interact and pull. We'll discuss a little bit more about how ATP specifically facilitates that. And so here we go. ATP and skeletal muscle contraction, and don't worry, there's an image on the next page that will highlight this. Now, we know there are 2 molecules in skeletal muscle. There is the actin protein, which is kind of it almost looks like a helical, is that a word? Yes, a helical fragment. And then there is the myosin protein, which is the protein that has the little moving heads on it, the actual parts that seem to contract. Now, actin has a compound, a protein around it called troponin, or troponin, however you want to pronounce it. This effectively is wrapped around the outside of the actin molecule and actually provides a place for the myosin head to bind. Now, as it stands, troponin is inactive. It doesn't bind to myosin. However, when a muscle receives an impulse, and the calcium actually travels to this protein. It exposes binding sites. Basically, if you think about it, calcium, when it flows in, activates or exposes the binding sites on actin for the myosin to connect to. So, myosin, which actually has ADP, which is adenosine triphosphate, and an extra phosphate attached to it, now, in the presence of calcium, reaches over and grabs onto the actin. As it grabs on, the protein, the myosin protein, actually changes shape and pulls along it, pulling these two proteins alongside each other and actually contracting. At this point, now remember, calcium has gone in, exposes the binding site, myosin grabs on and pulls. Okay, great. What happens at this point? Well, how do we get it off? Right now, this myosin head is actually stuck to this binding site. Nothing can happen. It's going to be stuck there indefinitely. What you actually need is ATP to cause the myosin to release and relax from this binding site. Otherwise, it's going to stay stuck there. As you can see in that last point, if anybody is familiar with the concept of rigor mortis, this is why it sets in. What happens in the case of rigor mortis? Well, what effectively happens is that in all the muscles, calcium starts leaking back and forth as all the cellular membranes start to break down. Calcium leaks into muscle cells, activates this troponin, exposes the binding sites. The myosin head reaches over, grabs on, pulls, holds tight, and then has no way of releasing because there's no more ATP left in the muscles. And now it's stuck and the muscle doesn't move. What happens though, ATP then comes in, attaches to the myosin, and the split of ATP, remember the full molecule attaches, and then that phosphate breaks off, which actually causes the myosin to release and go back to its ready position. Makes sense. So now you've got the myosin back where it started, you've got the actin right over there, and as long as there is still calcium present on the troponin, the myosin will keep reaching down, grabbing onto this exposed binding site of the actin, pulling along, ATP comes in, releases it, cocks back, goes over, pulls, keeps going, and the muscle keeps contracting. When there is no more calcium and the binding site is no longer exposed, the myosin releases and the muscle stops contracting and can relax. If there's no ATP, the muscle stays contracted and nothing happens. And there it is, visually speaking. Now, you can see that calcium is actually released. That's where it binds, and you can see that little connection right there. Myosin is on the lower part, and it's just kind of pulling along that actin repeatedly. That, in a nutshell, is how muscle contraction works. Obviously, as we discuss a little bit more on action potentials and nervous systems and everything else, and rate of twitch. We'll go into a lot more detail, but that's the fundamentals right there. So that is how ATP and calcium work together for muscle contraction. Now, let's talk a little bit about how we generate ATP in the first place. First system we know about, ATP, creatine phosphate. Everybody's familiar with creatine. At this point, most folks know via all the supplement companies that creatine phosphate is effectively an ATP storage molecule. Creatine phosphate itself has high energy phosphate bonded to the creatine molecule. And what happens is, as the amount of ADP rises in the muscle, creatine phosphate starts donating that extra phosphate to ADP, replenishing ATP. Now, these molecules, creatine phosphate, is not called in under normal conditions at an appreciable rate. One thing is important to note. All of these reactions, the production of ATP from ADP, the creation of creatine phosphate, the donation of phosphates from creatine to ADP to make ATP, all of these occur in equilibrium. If most folks remember from chemistry, thinking way back, almost every chemical reaction has substrates on one side and products on the other. And the equation is rarely a single arrow going in one direction. It is usually an arrow going in both directions, which shows that the compounds on one side and the compounds on the other side are in balance. There is always a balance between the two. Now, in many cases, there are a lot more products or a lot more molecules on one side than the other. So under normal cellular conditions, the amount of ATP, the amount of creatine phosphate that is actively donating to ADP is relatively low, but it still happens to a certain degree. That's important to keep under consideration here because no process in the body, or very few processes, ever occur solely in one direction. There are very few irreversible processes or processes that are not occurring in equilibrium. Regardless of that aside. So creatine phosphate, effectively, when it goes into the cell, does not have this phosphate molecule attached to it. It requires energy to create creatine phosphate, to donate that phosphate over there. So that can't be done when the cell is under active contraction. It needs to have a degree of energy surplus to generate more creatine. Now, as we said, everything is equilibrium. Every cell, every muscle cell has adenosine base, it has adenosine monophosphate, adenosine diphosphate, and adenosine triphosphate in it at all times. Now, when a cell is fully stocked and is balanced in energy, it has a lot of ATP, 10 times as much ATP as it does ADP. As the cell starts to go hypoxic and starts to be generating, and the energy utilization goes up, the amount of ADP rises and the amount of AMP rises. When that starts happening, the equation becomes favorable for creatine phosphate to start releasing its phosphate, and ADP starts grabbing those phosphates off it, regenerating more ATP. That happens faster and faster as metabolic needs on the cell rises. And as you can see, most cells have enough creatine phosphate to last for anywhere between 12 and 15 seconds of constant contraction. It is important to note no other substrates are needed. You don't need oxygen, you don't need glucose, you don't need the presence of any other energy substrate for this to occur. Of course, to resynthesize it, you do. You need significant energy substrates. You need oxygen. Muscular pH needs to be relatively alkaline. If it is too acidic, it will not regenerate. Moving on to energy systems. Now, continuing with energy systems. On the left is Usain Bolt, who probably needs no introduction. Obviously, extraordinarily fast 100-meter runner. On the right is CHP, Complete Human Performance Coach, and incredible ultra runner in his own right, Nicodemus Holland, who is, I believe, the youngest person ever to complete the Barkley Marathon, which is the infamous 100-plus mile, 57,000 feet of gain race that very, very few individuals has ever completed. Now, both these individuals are runners, of course, but each one has a physiology that favors a very, very different energy system. An individual like Nicodemus does not need the short-term power production of an individual like Usain Bolt, and Usain Bolt can probably not do anything for 50-plus hours without a couple meals and a couple sleeps in between. So, very different sorts of individuals competing in the same basic sport. They're both runners, but they're both not. Now, moving on to the actual energy systems that require macronutrients, let's talk about glucose and the processing of glucose to generate energy. Glucose has both an anaerobic and an aerobic method, pathway by which it is processed. So, in order to do anything with glucose, of course, the first thing we need to do is start breaking down glucose. Glucose itself has to be actually divided up into other molecules to actually become part of any energy generating process. First thing that happens is something called glycolysis. Now, glycolysis, or actually breaking glucose down, the initial breakdown, requires ATP. So you need to actually put energy in to get energy out. Of course, it only requires 2 ATP per molecule of glucose, so that's not a huge investment, but it actually results in several substrates, 2 pyruvates, 2 NADH, which we'll use later, and 2 net ATP. So it actually generates 4 ATP, but since it consumes 2, it generates 2 net. So it doesn't generate a huge amount of energy, but you also generate something called pyruvate. Now, the fate of pyruvate depends on whether or not there's actually oxygen present. If there is no oxygen present, we go on to the next energy system, which is what we call anaerobic glycolysis. So, It is also called fermentation. Now, fermentation takes place in many cells under many conditions. As you can see, it's the primary source of ATP in red blood cells, which obviously makes sense if you think about the fact that red blood cells don't actually have mitochondria. It generates very few ATP per glucose. So it is not the most efficient, or shall we say, cost efficient. You don't get the best mileage out of your glucose. Let's put it that way. That doesn't mean it's not a very quick process. It doesn't mean you can't survive on it well, but it just means that per molecule of glucose, it is not very effective. And as we'll see, there are a couple reasons why it is not, the preferred source of energy. So what does pyruvate do? Well, pyruvate actually breaks down into compounds called lactate and restores NAD+, which then goes around and, as you can see, actually allows glycolysis can continue. Remember, the initial process of glycolysis actually generates NADH, which means NAD+ goes into it. So basically, all that this does is it allows glycolysis to continue and allows ATP to continue to get produced. Now, NAD+ and NADH are actually major substrates in allowing this entire process to continue. And we'll see more of that later when we talk a little bit more about the biochemistry of the Krebs cycle. So what happens then? So we have pyruvate, and under anaerobic conditions with no oxygen, lactate is produced and NAD+ is generated. Well, what happens to it? Well, in muscle cells, there's actually evidence that lactate is slowly oxidized back to pyruvate. We used to think that lactate had to be shunted out of muscle cells and go back to the liver, and that was the only way to do anything with lactate. But as we're finding out, that's not actually the case. Once a muscle cell gets in oxygen, it can actually start to regenerate pyruvate from lactate. But we'll see more on that later. However, you can take lactate and take it back to the liver, and under a system called the Cori cycle, which we'll see in the next slide, we can actually reconvert this lactate into glucose. Of course, energy is required from this reaction. So you are actually utilizing ATP or using ATP. It is a net energy requiring process. This is an example of the Cori cycle. You can see on the right side, in the muscle cell, glucose is broken down into pyruvate through the process of glycolysis. And then from there, it is broken down to lactate, and a small amount of ATP is produced. Of course, you then take that lactate, bring it back to the liver, and lactate is converted back into pyruvate, and with the infusion of even more glucose, or even more ATP, becomes glucose. So you can see this is not the most energy efficient system, as there is a net massive loss of ATP. The advantage to this, of course, is that muscles can continue contracting, and if you have enough glucose, you can produce a tremendous amount of energy very, very, very quickly. Of course, it's not sustainable. This is a net energy loss from your glucose. So, not a great system, great short-term energy production. The aerobic pathway is significantly more complicated. The aerobic pathway, when there is enough oxygen to serve as a final oxygen acceptor, and we'll see what that means, what happens? Well, pyruvate is converted to a compound called acetyl-CoAce, which is at the very top of this cycle. Now, this takes place inside the mitochondria. It is important to note that anaerobic fermentation does not take place in the mitochondria. It takes place in the cytosol of the cell. So the amount of mitochondria has no bearing whatsoever on how much lactate your body, or how much glucose your body can actually process and convert to lactate, and how much ATP can be produced. So what happens in this cycle? Well, you can see at the top, pyruvate converted to acetyl-CoAce and pops into this cycle. And you can see that this cycle goes around and around and around. And every time it goes through, what happens? NADH, FADH2, and ATP are produced along with carbon dioxide. Now let's follow the cycle around a little bit. So acetyl-CoAce now converts to citrate, converts to isocitrate, and the next step from isocitrate to alpha-ketoglutarate, CO2 is produced, and NAD plus converts to NADH. Alpha-ketoglutarate to basically continue to follow through. I don't have to walk you through every single step, but you can see how much carbon dioxide is produced, and you can see that through various steps. Now, one of the things that's very interesting about this process, of course, is that it requires oxaloacetate to continue to come back through to combine with acetyl-CoAce to keep the cycle going. If there is either too much acetyl-CoAce or not enough oxaloacetate coming back in, this cycle does not operate properly. You need oxaloacetate to keep acetyl-CoAce going. And we'll see why this affects certain processes and what role this actually plays in the ketogenic diet and ketosis. Okay, so all of this is all well and good. What's actually going on here? This doesn't seem to be a huge way of producing ATP either. Well, next thing that happens is something called the electron transport chain. Because the most important thing that's continuously being produced here is large amounts of NADH and FADH2. Here is where it actually all comes together. Because the important production in the Krebs cycle, again, is not necessarily the ATP, it is all the NADH and FADH. Now, here is a great example of how channel proteins could actually be used to generate more ATP. The electron transport chain is actually a way of generating a gradient across the mitochondrial membrane. So what happens is NADH and FADH2 are all used to help continue to create a gradient that pumps hydrogen ions outside. Well, hydrogen ions are also just known as protons, to pump those outside of one part of the membrane. What happens then is those ions flow back in, and in the process of flowing back in, they pass through this compound, they pass through ATP synthase, this particular protein. And in the process, that energy, I mean, effectively, if you think about it, all these protons getting pumped out are creating electrical gradient. That's that electron transport chain. They flow back in, and that potential energy is used to resynthesize ATP. Now, when we talk about biochemistry, we'll talk a lot more about the specifics of this, but that's really what you need to know here. Now, oxygen is actually utilized here as a terminal electron acceptor. What does that mean? Well, oxygen is very high potential energy. And as these things flow through, you can see hydrogen ions and electrons. The byproduct of this is water. Without oxygen, the electron transport chain does not operate. And again, we'll talk about more specifics of this later. So we can see in this entire process, what is produced? Well, glucose is consumed, carbon dioxide is produced in the Krebs cycle, and oxygen is needed as the acceptor in the electron transport chain, which is how a huge amount of ATP is made. Next portion of the energy systems. Well, how about fat? Where does fat come into in all this? Well, fat metabolism is what we call beta oxidation. That is how a fat molecule is broken down and actually enters into this system. Now, fats, if you are familiar with the structure, and again, we'll go into this more later, are essentially long carbon chains. Every single carbon is basically can be broken off one by one, and through this entire process as outlined here, effectively becomes acetyl-CoAce. What does that mean? It basically means if you think about fats, a fat molecule can be thought of as a very long chain, very slowly broken down acetyl-CoAce donor. And as we saw earlier, acetyl-CoAce becomes part of the Krebs cycle. So if we think about it, whereas glucose is quickly broken down and just dumps into the Krebs cycle, fats are very slowly broken down via beta oxidation and become acetyl-CoAce. Now, beta oxidation of fats, it's so called because of the process of breakdown, takes place in the cytosol as well. It's then all those compounds, as you see, the fatty acid is broken down to fatty acyl-CoAce, which is combined with carnitine. That is transported to the mitochondria, where fatty acyl-carnitine is broken down to fatty acyl-CoAce, which is then broken down into acetyl-CoAce. Now, initial beta oxidation does not require a mitochondria, but the breakdown of the product of that beta oxidation does take place in the mitochondria. So more mitochondria means that more of these substrates can actually be converted into energy. Now we get to ketosis. Ketosis will be its own segment when we get to nutrition. But fundamentally, what is ketosis? What induces ketosis? Well, ketosis is induced when acetyl-CoAse levels rise very high, in part because of a high amount of fat, but oxaloacetate, which is actually, if you remember back to the Krebs cycle, the last compound that's combined with acetyl-CoAse to keep it going. Oxaloacetate, now the Krebs cycle, what's interesting about it is the Krebs cycle doesn't just exist as a cycle on its own. Many of the compounds that are intermediaries in the Krebs cycle can be utilized for other needs in the body, or other compounds, these compounds can be generated elsewhere and get inserted into the Krebs cycle where they're needed. So it's not a closed cycle. The Krebs cycle is a cycle where various intermediates can be grabbed and pulled out and put back in. In a case where glucose levels are very, very low and glycogen levels start to get depleted, oxaloacetate, which is one of the intermediaries, or the second to last compound in the Krebs cycle, is pulled out via gluconeogenesis and consistently removed from the Krebs cycle at that point to generate glucose and glycogen, which is used elsewhere in the body. Remember, everything takes place in a balance. Every reaction in the body is attempting to maintain homeostasis. You can't just continuously deplete glucose and deplete glycogen stores and expect the body not to try replenishing them, even if the body is under stress and trying to utilize these substrates. If you get high acetyl-CoAse because of endless beta-oxidation of fats, but very low oxaloacetate because of very high glucose needs, because glucose intake or carbohydrate intake is very low, what starts to happen? Well, in the liver, acetyl-CoAse starts to go down this separate ketone body generating pathway. The liver is the only place where this takes place. And what happens is acetyl-CoAse is processed by the liver into these ketone bodies, where they are then transported around the body for utilization and insertion into the Krebs cycle. Again, we'll look at this more later. Now, important to remember, and one point we're just going to touch on here, this is not the same thing as ketoacidosis. The production of ketone bodies is responsible, yes, for ketoacidosis, but it is the runaway production of ketone bodies that leads to ketoacidosis. This will be very, very relevant later on in the nutrition segments. Now, these ketones, as they are released from the liver, they travel to cells, they produce NADH, and they enter the Krebs cycle as acetyl-CoAse. In a nutshell, that's all ketosis is. Low oxaloacetate because of glucose needs, high acetyl-CoAse because of the presence of fat, which, if we think about it, is pretty consistent with ketogenic diets. So those are the basic ways in which your body can generate ATP. Let's take a look at a quick practical application here. Now, when we talk about energy systems, let's think about, well, okay, so we know about lactate, we know about the lactate production. What is the difference between that, or when does the body decide, what is considered a lactate threshold? What is lactate? How is it used in training? All right. Well, one of the biggest, kind of parameters when we talk about aerobic performance would be the lactate threshold. Now, sometimes people talk about VO2 max. We have lactate threshold, we have VO2 max. How does all this relate to energy systems? Well, VO2 max actually has very little to do with energy systems as we understand it. VO2 max is simply, as noted here, peak oxygen utilization by body over time. Now, VO2 max, as expressed as a relation to body weight, is milliliters of oxygen utilized by kilogram per minute. If you think about it, obviously a larger individual can probably use more oxygen, but they can't necessarily use more oxygen per kilogram of body weight. You are very, very unlikely to hear a large relative VO2 max being expressed by extremely large individuals. Why is that? Well, their absolute oxygen utilization will be higher, but their relative oxygen utilization may not be. There are certain rate-limiting steps in oxygen utilization. As an individual gets bigger and accrues more muscle mass, they may use more oxygen overall, but the system by which oxygen is transported to their muscles isn't necessarily growing. As an individual gets larger and larger, They are getting oxygen into their system, but their heart is not necessarily growing at a linear rate. Their lungs are not increasing in cross-sectional or surface area at a linear rate. As individuals get larger, even the most efficient individual will notice that their VO2 max may not increase linearly. So VO2 max is a good proxy for aerobic performance, but if it just refers to oxygen utilization, is this necessarily referring to how much productive work the individual can do? Well, it certainly refers to the maximum amount of oxygen they utilize at any given time, but does that mean they are necessarily performing well over time? The individuals who are typically associated with peak short-term performance, like sprinters, may not have a very high VO2 max at all because it doesn't matter. Remember, ATP, creatine, phosphate don't require any oxygen whatsoever. If you have enough creatine to last for 10 to 15 seconds of contraction, if you think about it, the 100-meter dash doesn't need any oxygen whatsoever. This is taking place under 10 seconds at the world-class level. You don't even technically need to breathe at all during a 100-meter dash because your muscles have all of the substrates they need to contract fully for the entire time period. Now, your muscles may not need the oxygen, but your brain probably does. So most 100-meter runners will continue breathing just a little bit to ensure that they don't pass out at the end of the 100 meters, but this is not an oxygen-requiring activity. Peak VO2 max does not matter for a 100-meter dash. It does not matter for a 50-yard dash or a 40. It does not matter for a maximum set of squats. These are all unrelated to your VO2 max whatsoever. VO2 max matters more and more as duration increases, but again, VO2 max only represents an absolute peak oxygen utilization. It doesn't measure actual practical, sustained oxygen utilization or output. For that, people talk about lactate threshold. Now, lactate, as we talked about, this anaerobic system, lactate production is something that takes place again outside the mitochondria, and it used to be considered just a waste product, a completely useless byproduct of anaerobic fermentation. That's not actually the case. They're finding more and more that lactate itself is actually an energy substrate, but this is all still theoretical. We won't discuss that too much. But what is important to note is that the degree, remember how inefficient lactate is as an energy substrate. Each bit of glucose produces very, very little energy in the lactate system. So the longer the body can actually go before dipping heavily into the lactate system, the higher degree of intensity and duration the body can go at before dipping into that system, the better. So what is the limiting factor here? Well, to avoid going down into the anaerobic fermentation process, the body needs to be able to get as much oxygen to the muscles as possible and needs to continue, of course, clearing out that lactate at the same time. Why? Because again, everything is in equilibrium. If the body is producing too much lactate, it's not actually, as we're finding now, the runaway levels of lactate that are related to poor performance. It is simply the fact that the body is going so heavily and tapping so heavily into the system that is correlated with a decrease in performance. So it's not lactate itself that shows that the athlete is at their peak level or performance, or that's as far as they can go. It is simply that lactate or high lactate levels and high anaerobic fermentation, the body dipping into this process is simply showing that muscle cells and the athlete themselves are at their limits of performance. So it's correlated, it's not causal. It's not that high lactate is associated with a limited performance. It is that utilizing that system heavily shows that the body is no longer able to sustain aerobic glycolysis. Lactate threshold is basically the point at which lactate levels rise above their resting levels. Now, that seems like it would be pretty much anything beyond just kind of sitting on your butt. However, lactate levels start to rise only noticeably when activity levels hit anywhere between 75 and 80% of VO2 max, or max heart rate, whichever you want to go by. Now, what does this mean? Well, that 75 to 85%, that's already a range. The lactate threshold, the point at which lactate levels start to rise, which indicates that the body is not able to continue getting enough oxygen and not able to carry out sustainable peak energy production through the Krebs cycle, as an individual gets more and more fit, as the number of mitochondria increase, as the blood capillarization, the amount of circulation to the muscles increases, the body can utilize more and more oxygen by the muscles. The body can carry out more and more, a greater and greater amount of aerobic glycolysis. It will have more oxygen to serve as terminal acceptors in the electron transport chain. As an individual gets more fit, their ability to utilize aerobic glycolysis at a higher level of intensity increases. So a very fit individual may have a lactate threshold that is only slightly under their VO2 max. In other words, their ability to utilize, to go through aerobic glycolysis may occur at a very high rate of intensity simply because they can not only utilize massive amounts of oxygen, but they can continue to do it repeatedly because they have so many mitochondria, because they have such good circulation. A relatively novice athlete, even if they have a relatively high VO2 max as indicated by a transient ability to utilize oxygen, if they can't sustain it, if they don't have the capillary perfusion, if they don't have the amount of mitochondria that can sustain this for an extended period of time, and if in other words their system is very much a short-term, I don't want to say one-trick pony, but if they aren't able to sustain performance, then their lactate threshold will be relatively low. Now, does this mean anything, again, for a sprinter or power athlete? Absolutely not. In fact, it doesn't make any difference whatsoever. Again, a sprinter, VO2 max doesn't matter. Lactate threshold doesn't even really matter. Again, not a relevant energy system. As you get to longer and longer duration athletes, however, a higher lactate threshold simply shows that the individual has the needed infrastructure in their body to operate at a higher level of intensity. Another thing you may hear about is OBLA, or onset of blood lactate accumulation. That is simply an indication that the body is at 4 millimoles per liter of lactate. It is an absolutely, I don't want to say it's an arbitrary number, but that is kind of considered experimentally to be almost the point of no return for lactate accumulation. So what does all this mean when we talk about athletes and measuring their lactate threshold? Well, why do we want to measure someone's lactate threshold or VO2 max? Well, does it matter for their sport? Again, for sprinters, it doesn't even really matter. What's the point of measuring VO2 max if VO2 max is not a limiting factor in performance? Well, a lot of these things are correlated with improvements of performance that can indicate improved physiological adaptations that can support their sport or activity. A higher lactate threshold can be correlated with improvements in cardiac stroke volume, improvements in respiration, improvements in perfusion, improvements in mitochondrial density. A lot of these things are correlated with overall improvements in athletic performance. Now, for an individual like maybe a 10K runner or a half marathoner, obviously the higher the VO2 max and lactate threshold, the better. Because that simply indicates that the athlete can perform at a higher and higher level of relative intensity. However, if an individual with a high lactate threshold and high VO2 max is still slower over time than an individual with lower numbers, it clearly means something is wrong. Remember, these are not absolute numbers. A higher VO2 max or higher lactate threshold does not indicate better performance between two athletes. These should only be used as individual metrics of performance increase. VO2 max represents an absolute potential for that athlete. Lactate threshold, increasing their lactate threshold, indicates that the athlete has gone through adaptations that are going to allow them to perform at a higher level of intensity for a longer period of time. It does not mean that one athlete is necessarily superior to another, because as we've indicated, all of these things are absolutely individual. And again, much like in lifting weights, much like in resistance training, where skeletal muscle strength is not necessarily, or absolute contractile strength is not necessarily indicated with even being able to lift more weight than another individual. These are internal metrics for measuring performance relative to themselves. That is the basic primer on energy systems, and if there is one important takeaway that I want to continue to emphasize, it is that every energy system is being utilized at all times. Now, remember, ATP and CP. Remember, adenosine monophosphate, diphosphate, triphosphate, and creatine phosphate are always in equilibrium. If an individual is running at very, very low intensity, they may not utilize much creatine phosphate, but they are still producing some ADP. Once that 10-to-1 ratio of ATP to ADP starts to increase even to 9.9 to 1, the body is still utilizing some creatine phosphate, even if it's on a very, very reduced scale. It's not an absolute system, so you could say even at a slow walk, your muscles are utilizing some creatine phosphate. Even during a 6-second sprint, on the other hand, rate of beta oxidation of fats increases very slightly. Why? Because energy demands are going up. So all the various bodily processes that metabolize fats, that release fats from your stores, get the fats to cells, transport them, bring them into the cytosol, get them broken down by beta oxidation, get those products of beta oxidation to the mitochondria, get those products further broken down, get them broken down in pyruvate, put them through the Krebs cycle, put those products through electron transport chain. That is still happening even in a 6-second sprint. It is not a major contributor to performance in that sprint, but it is still taking place. Again, all of these energy systems are being utilized at all times. That is the wrap up on Introduction to Energy Systems. Thank you very much.